this afternoon session, this public session of the kickoff of the project AtlantECO. I want to welcome all the people that is listening or participating to this, even if they are not participating directly to the project. This is a large-scale project on the Atlantic, as you know, on the assessment and prediction of the status of the Atlantic, of their ecosystem services, and on the future provision of them, as I will tell in a moment. I'm just happy to say that it will be a huge adventure because we are going to have this truly multidisciplinary effort, but also it will be an adventure because we are going to have true expeditions going in some of the wildest places of this planet. I'm sorry because of the COVID situation, we had to have this virtual, not in person, and that obliged to readjust and reorganize many aspects of the project, and in particular this meeting, but we cannot do anything about it. And so it is a research project. It should produce new knowledge on this ocean, but also should produce cooperation, enhanced cooperation. This is really important. I will talk about this more, but the importance of cooperation motivated us to have within this public session some other person talking from South Africa, from Brazil, from US, from Canada, to talk about the international context of the research at the moment, and to enhance synergies in the future, and to enhance the, let's say, the links with the present international context of research, not only research in this field. Let me start with this a short project overview, or I'll try to give you the generally, basically the main idea of the project and a little bit about the background, and then I leave the work package readers to really actually present the activities of the project. So, you have to allow me to proceed.
You can do it now with this.
So, of course, we are going to talk about the Atlantic Ocean. And needless to say, that is a very important ocean basin for many, many reasons. The role in the Earth system, the very different rich ecosystems that characterize it, but also the important value for the economy of the countries that are around the basin. That said, we know that there are many ocean health issues and also issues related to the sustainability of those activities, which should be addressed. But also, it's also important to underline there are a lot of large inequalities between countries, but also within countries, inequalities of any kind. So, in this context, the general context is, you know, we want to have a better management of these natural resources, and the best is to have a virtual circular loop between the observation of the status, the understanding of the processes that produce modeling and informs policymakers that can act through government and to improve the health of the system. The problem is we have to inform with knowledge this process, so that otherwise it doesn't work. And as we know, for regarding marine ecosystems, there are a lot of open questions, especially because it's a complex system. So a lot of research is required. So our aim is essentially to contribute to feed this kind of circular loop. What is also important to keep in mind that there are no more local problems. Problems are almost always global because everything is interlinked. Of course, the ocean is interlinked because of the interconnection given by the ocean currents, but it is also the economies are now linked. So here you see, for instance, that basically there is a strong link between the fisheries in Brazil and in tropical Africa, and actually Europe and the United States. So any impact of the climate change on those countries will have a direct input on these more northern countries in terms of provision of proteins, and this will feed back on the economies there. So, so in this, within this general context, the European Commission decided to launch a series, a flagship, an All Atlantic Ocean Research Alliance flagship program, which has funded a series of projects like our one, with the aim of having a systematic approach to tackle the scientific and social economic challenges and to move towards a basin-wide cooperation. More specifically, the aim of this action, which is now described in the balance statement between European Community, South Africa and Brazil, is that the specific aims are to assess the status of the ecosystem and the ecosystem services, to predict the future trajectories, to improve sustainability, reduce inequalities, and to actually create ocean-engaged citizens through literacy. So we set out consortium, particularly to this goal. We were successful. So now we have this large-scale consortium made of 36 partners for several countries, with a budget of about 11 million euros, and the duration of our project will be 4 years at the moment, with the starting date the 1st of September. Our vision, we have a very young attendee. Our vision is to develop an holistic framework to manage the ecosystem health and services, and to provide knowledge-based resources, so to include knowledge behind the resources that are made available to science, industry, policy makers, and citizens to manage. And last but not least, the exchange and build capacity along and across the entire Atlantic Ocean. The project is built around 3 main pillars. The first, scientific research pillars, let's call it. So the first one is the oceanic microbiome. So the microbiome is this invisible majority that lives in the water, in the oceans. It's called also plankton, and is made of everything that is invisible or almost invisible. So viruses, phytoplankton, bacteria, archaea, and zooplankton. But when we say microbiome, is that we assume that all those things are interacting. So it's a unique ecosystem made by many actors that are interacting between them. So microbiome is important, as it happens for the humans, for the gut microbiome. It is very important for the functioning of the systems. It actually makes 80% of the biomass of the water column, and regulates many ecosystem services, from climate, harbors several pathogens and parasites. Think about aquaculture. It channels pollutants, including plastics, and supplies biomass. Ultimately, all the seafood comes from the microbiome, and also it's important that is being very sensitive to the environmental condition. It actually creates a link between climate variability or climate change and the higher trophic levels, like fishes. So we in this effort, in working on this microbiome, we are going to build on a pioneering effort, which was a Tara Ocean Expedition. Tara Ocean Expedition is pioneering this phenomic observation, that is to put together the observation of the organism using advanced microscopic tools, together with the sequencing, the analysis of the genomic and genetic properties, so their DNA, their RNA, and these are the aim to better understand the functions, and also to have a better characterization of who was there at the moment of the sampling. But also the other important pioneering effort is to put together a vastly multidisciplinary consortium that had 10 years to develop an approach that is really fully merged from physical oceanography to molecular biology. Building upon this effort, we are now starting this new adventure, which is like merging the oceanographic community in a sense with community working on advanced biology. And this is can be sound fancy, but in fact is actually what's happening in the human health research. So this is the case for COVID-19. COVID-19, the understanding of the symptoms is actually going through the same exercise we are doing. So basically sequencing the DNA and RNA of patients, trying to understand what kind of alteration of the metabolic state is causing this ill state. So this is a bad health state. Basically, what we are going to do is do the same in the ocean. So using genomics and data science together to create new knowledge, to produce new understanding. The challenge we have is this, because we start from the large scale view of the system, so biogeochemical scales, the global ocean, but then if we really want to understand and predict the functioning, we have to go through the community, so smaller size, so the interaction between the organisms, but also we have to go down to the molecules and the metabolic function at the gene level to really understand why we have a specific response to climate change, for instance. This means also to put together a lot of different disciplines, which is why not only nested complex system, but also the need to create a new approach, a scientific approach. The aim in that would be to create this sort of circular path of information, a flow of information from the measurement at sea to the laboratories, to the modeling, to the data science that should produce synthesis and knowledge, and then back to the measurements. Without this connection, we cannot really understand the system. To say this, that's why we created a very multidisciplinary, actually young, global team. We have 29 countries represented here, and I'm very happy to say that Brazil and South Africa are really nicely represented in this project. And then we have relatively, I would say, young people with different backgrounds, different roles, and some contribution from private sector and third sector. The second pillar is a seascape. The seascape that is a connectivity. Everything is on the move in the ocean. The nutrients moves, the organism moves, pollutant moves. So we have to. So if you want to understand what happens in one specific place, you have to go back in time in the trajectory where the water was coming from to really understand. So connectivity is crucial to really address the question you want to address. But it's also important that to note, as you can see on the right side, that actually circulation is also a stressor. So a change in the circulation can have a deep alteration in the ecosystem status, for instance. Think about climate change. A third pillar is microplastics. So microplastics, you know about that, so I'm not going to discuss, and later on we'll know more, learn more. Just to say that there are many processes that actually, and many sources that actually rule and regulate the distribution of microplastics in the ocean. So we are going to focus on some of them, like rivers, sources, transport in the ocean. And it's a long way to go, but we think we can really contribute to that. And most importantly, we want you, instead, as usually done, having microplastic focus separated from the rest, we are going to do this as a unique question together with the ocean microbiome. There are many reasons for that. The main one is that there is a lot of life on plastics in the ocean. Some of those guys are actually toxic, algae, other are bacteria, and they rule also transport of the plastic in the water. So in short, we use the same tools, the same approaches we are going to use to characterize microbiome, to characterize who is living on the plastic, and to have a better understanding of the processes regulating plastics distribution in the water, but also the potential impact, for instance, on ecosystem services. So the project is organized around 4 activity streams to address the main question. So the first is to assess the status of the ecosystem structure, their functioning, the health, and also the services provided. So we hope to move from this beautiful map from Benjamin Franklin to maps like the one on the right, which is made using the genomic information. So the map on the right is one of the first biogeographies based on the analysis of the DNA of the microbiome. Second exercise, a stream of activity is a very important one, is to enhance knowledge and innovate. So basically we'll move forward in terms of adding protocols and tools for the observation. We will apply artificial intelligence and network analysis to extract knowledge from the data. We will have improved connectivity and biogeochemical models for predictions. And nicely, we used to do expedition or design, new expeditions. Third activity streams is to identify the drivers and stressors of the system and forecast the future of them. So we will focus on both the ecosystem drivers intended as a climate change, change in storminess, extreme events, heat waves, but also in terms of socio-economic stressors. So what is important is that we are going to consider this multiple stressor approach. That means that sometimes more than one stressor is actually acting on a single place. Like here on the right, we see fishery boats together with the plastics in the same place. So basically, two stressors acting on the same ecosystem. Finally, the fourth activity stream would be to share and use capacity and knowledge. This means to organize capacity building internally through training, workshops, and other linear process, is to inform policymakers to activate this flow of information between response to a problem, identification of the problem, assessment, but it means also to reduce inequalities. Inequalities, which is one of the goals of the Sustainable Development by UN, it will be one also of our goals. Finally, we'll push and disseminate a frugal science approach to allow as many as possible students, also in secondary schools and other situations, to start doing some science on the microbiome. These four activity streams are actually organizing the overarching structure that organizes our packages that will be described later on, and which describe what I just mentioned. I'm not going to talk about them now. Ecosystem services, we are going to deal at least from the beginning, then maybe we'll add some more, but at the moment we are going to focus on the carbon drawdown, which is the classical one, on the vertical transfer of matter and food from the surface to the mesopelagic and seabed ecosystem, on aquaculture and fisheries through a lot of collaborations, and also cycling on plastics and biodiscovery. In some cases, these are new activities, and some are very well established, like fisheries as an ecosystem service, or carbon drawdown as a climate regulation service. But in other cases, to relate the microbiome to the services provided by the ocean, it's not an easy task. It will be one of our challenges. We will favor internal discussion animation to allow for a truly multidisciplinary exercise. So we identified 5 or 6 initial thematic groups that should work together, cross work packages to advance on the most challenging concepts and tools we want to apply and use. This will be made through scientific animation, webinars, workshops, summer schools. One of the most challenging one will be indeed to apply artificial intelligence and theories from complex systems to oceanography and ecology. We will have some case studies which actually are thought to bring us toward applications, so to prepare for the use of our results. A few of them, in short, I mean, the first is basically to work with people working in agriculture that are preparing to the sea level rise, which means the increase of salinity in the soils. So they want to work with us to analyze, to see if we can improve the microbiome of potatoes, in this case, to make them adapted to this increase of salinity in the soil. Then we work with industries to check the impact of drilling in Brazil and to improve policies for environmental protection. We work with aquaculture projects to improve the early hazard detection, things about parasites, harmful algae. Then we work in South Africa with the ministry and with the local actors, private actors, to improve the regulation for improving the environmental protection because of the shell of semi-diamond mining. And finally, we work on improving value chains for food production assessment with the Oxfam NGO. To finish, we will work through synergies. First of all, we have to recognize that AtlantECO occupies a well-defined niche, working on the microbiome of the invisible majority, It's basically, and coupling advanced genomics with modeling and data science and physics. It's basically a unique project at the moment. So it's somehow very natural to establish connection with projects working on fisheries or on climate variability and so on and so forth. We will have as a general framework this anchor action, coordination action, which will help us, as Sophia will tell in a minute, to coordinate and to have a more efficient exercise. But most importantly, there are at least 6 up to 10 other projects that were funded at the same time by European community, so of similar size as us. So we are already working actively to establish synergies with them. And then there will be synergies with other countries, national programs, JPI Oceans, and with cruises to enhance the geographical coverage of our sampling. Thank you very much. So now we will have an overview of each work package, of their main goals, challenges, and activities. And we will start with the first work package 2.
Thank you for your attention, for being here. Thanks, Daniele, for the nice introduction. So I'm presenting work package 2 that I co-lead with Mikey Vogt from the Swiss Federal Institutes of Technology in Zurich, and I am from the University of São Carlos in Brazil. So this work package will deal with the marine ecosystem structure and function. Let me see the slide. Okay. So in this work package, we'll try to compile all the data available on the Atlantic Ocean. So you can see my mouse, right?
Can you just let it go just for a second? I need to do something.
Okay. Can I point with the mouse? Yes. Okay. So in this first map, you see the existing observations on microbiomes. You see there are lots of them, mostly in the North Atlantic, not so many in the South. That's one of the gaps that we will try to fill in this project. Then in the map B, you see observation that exists already on plastics and plastisphere. And then in the figure, in the map C, you see observations in carbon floods. Okay, so we'll try to merge all these data sets. Of course, all these cruises generate lots of heterogeneous data, so lots of different sources of data, from microscopy to pigments to microbiome genetic data. And it's not always easy to merge all these data sets. They are usually separated in different publications from one cruise, and then this data stands on these databases, but they're not in a single database. It doesn't exist, and most of all, we don't have yet a georeference database with all this data. So that's one effort we would like to do in this project. Okay, so we have all this kind of data. And also we have this new problem of the plastics that we also need to address. So let me see. Okay. So from these expeditions, you get tables of data. That's what's in this picture. That can be image data, can be molecular data, DNA from genetic analysis. It can be plastics data like images, or even microorganisms that live on plastics, and also all the metadata or physical data from temperature, salinity, everything that you get from oceanographic cruises. So our work package 2 will be to gather all the observations that exist in Atlantic, or organize all these tables and put coordinates, and then produce maps. Okay. These kind of maps for several different things, from bacteria, for functions, for plastics, everything you can imagine. And then we'll try to feed our models with this data. Okay, so that will be our main activities. So to gather all this data, to produce maps, And then we'll try to demonstrate that this data has potential for biotechnology by bioprospecting and so on, and also turn this database available for the blue economy stakeholders. Okay. So resuming all this work package, we'll try to access and make a picture of the Atlantic Ocean structure and function. So the team is a very large team, lots of partners contributing to this work package. We only put the pictures of the leaders of the work package. So Mikey and Fabio is postdoc, and then I have in my lab Pedro and Paulo in the picture, but there are many people contributing to this work package. So it's a team of highly skilled oceanographers, modelers, and industrial. There's also SynTef will work with the bioprospecting. Okay, so we hopefully provide this database and also increase our database with new observations produced by the project and incorporate it into our database. That's what working package 2 is supposed to do. I think I'm on time. Thank you. Okay, there's, oh, I finished.
I'm just taking back the control.
Okay, sorry.
Okay.
If you have any questions, I'm not sure it's now, but
I think we have a question and answer session at the end, and if people want to submit their questions through the Q&A panel, then please feel free. Move to work package 3, and I will give control to someone.
Thank you. I'm trying to see if it's working. Good afternoon. Yeah, is it working? Okay. So I'm Simone Libralato. I am working at the National Institute of Oceanography and Applied Geophysics in Italy, and I'm co-leading the Work Package 3 with Sebastian Villasante from University of Santiago de Compostela. The Work Package 3 is about marine ecosystem health and services, and what is the main challenge that is considered in this work package is actually the challenge of integrating the large set of information that are going to be acquired with the project, but also with the sister projects and also with other kind of available data sets. The challenge is to capture the human-ocean coupled system, so integrate the multidisciplinary information into something that can evaluate also the services. And we have a series of information of different kind that will be integrated to evaluate and quantify the health of the ecosystem. To do that, we will use an already available framework that is an Ocean Health Index. Is a framework that is allowing us to evaluate under the achievement of current societal goals and services the health of the ocean, the Atlantic Ocean. I cannot go on. Oh yeah, here. And we actually, oh sorry. The Ocean Health Index is resulting from a work done by Halpern and colleagues and published in Nature in 2012, and is actually representing the different activities and services provided by the ocean in 10 global goals that goes from food provision to artisanal fishing opportunities, natural products, carbon storage, coastal protection, tourism, recreation, economy, sense of place, clean waters, and biodiversity. With the AtlantECO, we are going to tackle especially some ecosystem services that are here highlighted, but because of the properties, of the important properties of the microbiome and the microplastics in the ocean, these are actually embedded in other services. Any one of these goals, in fact, is a result, the evaluation of each goal is a result of status, pressures, and the resilience factors that are many layers that we are going to integrate, and where the microbiome and other results coming from AtlantECO will be included. So it is expected that we are going to evaluate several ecosystem services using these goals of the Ocean Health Index, but also assess the impacts of the microbiome, of the plastic spheres, of the connectivity on other services. The team involved is very large in terms of expertise. There are 8 institutions involved, with experts going from ecological modelers to biogeochemists, from fisheries experts, economists, benthic ecologists, and data scientists, but also marine microbiologists. All these people is actually, this multidisciplinary team is involved in this integration, bringing expertise in order to provide the best assessment of what is the health of the ocean using these indicators. We will use the support of the developers of the Ocean Health Index. In particular, we are going to have the support of Ben Halpern, that is now the director of University Santa Barbara in California, and at INSEAS, and Katie Longo, this is a senior scientist now working at Marine Stewardship Council. These 2 experts will help us to remake the assessment using the Ocean Health Index and to integrate all the information that we are going to assess with the AtlantECO. The activities of the work package 3 are divided into a first assessment of the ocean health of the Atlantic, integrating the new knowledge on microbiome, microplastic, circulation, productivity especially, but using results from models and data collected. There will be a great effort in updating, integrating, but also advancing, because the new assessments will try also to incorporate the new omics data that will be acquired using also AtlantECO and the previous activities. The idea is to perform the assessment at the level of the Atlantic, but also focalizing in some particular areas, making regional calculations. And there is a second activity devoted to the socio-cultural and economic evaluation of ecosystem services. This will be particularly focused on carbon sequestration and provisioning services, fisheries and aquaculture. This is a socio-cultural evaluation that will be done for evaluating the effect of protecting biodiversity and generating these ecosystem services, and how these conservation will help to human well-being. The third activity of the project is interacting with existing European marine strategies. So these activities will identify links and dependencies between what are the Ocean Health Index goals that were presented before, and different kind of strategies and activities going on that include also the essential oceanographic variables or biodiversity essential variables, but also marine strategies framework directive descriptors that will be compared and associate with the Ocean Health Index dimensions in order to identify overlaps, gaps, and trade-offs. And synergies in the use of these indicators to evaluate the health and the future of the ecosystem changes. All these activities will be done in the very large integration with several other work packages, and also there will be results produced by these work packages that will feed for example, the future the work package 8, where there are the forecasting of the future changes in the ecosystem health. And I think that's all for now. Thank you very much.
We're now going to work package 4, and I will pass the control on to Stefan.
Let's check. We go with the mouse. Okay. Hi everyone. Augmented observations and technological innovations are led by Rubens Lopez from USP in Brazil and by myself, Stéphane Pesin, at Humble EBI. There we go. So the challenge of our work package is that there are lots of gaps in our knowledge base. Gaps can be spatial. We know that we have a lot more data, a lot more information in the north compared to the South Atlantic. It's also on the vertical. We have lots more data at the surface of the ocean than in the ocean depth. So gaps are also temporal. We tend to sample at timescales that do not always match the timescales of the processes that we study. So that needs to be fixed. Gaps can be ecological. We also tend to focus on the most abundant chemical or biological entities. We need to look at the full spectrum of these entities. And the gaps can also be technological in the sense that many innovations are on our doorstep, but we need to pick them up. We need to apply them in our activities. Oops, click again. Okay. But the good news is that it is actually the right time to fill these gaps. There's lots of momentum created by the Galway and the Benham statements, also the UN Decade of Ocean Science. And so that results into many initiatives. Many of them are going out at sea to collect new data. So it is the right time to fill the gaps. So our goals are to build capacity around standard protocols and best practices, to generate new observations with targeted sampling activities, and to innovate with high resolution sensors. And to do that, we have several activities in Work Package 4, and also a large team of partners that are expert in what they're doing. So let's go through the activities. The first one is on standards and best practices. That's led by EBI. What we will do is organize workshops. The first one will happen in November this year. And each workshop will lead to a handbook of standards and best practices. And we'll have a new edition every year. So a new workshop and a new version of that handbook. So we build on experience during the project. And we involve many people, not only people from the consortium in these workshops, we also open to other projects, other initiatives like MBON and Goose. And we also want to open even more to the community. That works that way. So we, together with AORA, AtlantECO launched earlier this month a special issue in Frontiers that targets, so we'll accept papers that show how they do, what methods do they use to sample and analyze microbiomes, and mainly microbiomes. Then, for most activities in Work Package 4, we adopted the hub and nodes approach. We did that because it is a main goal of AtlantECO and other projects like this, to share capacity and transfer know-how about how metadata should be collected and preserved and organized, how samples should be biobanked, how genetic data should be analyzed, and the same with imaging data. So we have a hub composed of key partners. We have EMBRC, Socio-Ecological, Humble EBI, Sorbonne Université, and World Courier for the shipping of all the samples. And their goal is to harmonize the methods in all the regions and ensure quality of the analyses. And that is spread towards nodes, regional nodes in Europe, in South Africa, and in South America. And we are also in discussion with Canada and the US to perhaps identify nodes in those countries so that we can be full Atlantic in that approach. So in terms of activities, we have several flagships in AtlantECO. First category of flagships is with oceanographic vessels. So we will go on board the Discovery in the James Cook for the Atlantic Meridional Transect. That's the yellow line, coordinated by PML. We'll go on board the Alpha Crucis coordinated by USP in Brazil to look at the South Brazilian bight. And we go on board Atlas, not Atlas, but Agulhas 2 with the South African partners to study the Good Hope line and to focus on process studies and experiments. And we also link up with external expeditions like exports from NASA, OSNAP from UK, GoShip from the US, and Polarpod from France. The other categories of flagships, we have research sailboats. So we have 3 of them. Tara will do the mission microbiomes, going along the north of South America, crossing the Atlantic through the gyre, and then going to Cape Town, and then going up the coast of Africa. So all these flagships will apply the standard protocols that we discuss and agree on during the workshops. The second ship is the Jürgen Seibold from the Max Planck Institute. They will also do the sampling, but in the North Atlantic. And then the Valerio Eco from University of Santa Catarina will sample along the coast of Brazil. And you will notice that for the Eco, both the Eco and Tara will sample rivers, the R's. So that's important. 5 rivers in South America, 5 rivers in Africa, so that we sample microbiomes, plastics, close to the cities, along the rivers, and going out into the ocean. Then we have activities with marine stations and with citizens. You probably heard about the Ocean Sampling Day, the OSD. The original network of OSD is mainly the blue and the black stars on the map. So in 2022-23, we will organize an all-Atlantic Ocean Sampling Day by extending that and adding marine stations along the coast of Africa and South America. And that's coordinated by EMBRC and HCMR for AMBRC. Then with citizens, we'll do the citizen Sail for Science. We build on the initiative Plankton Planet that generated amazing results in 2015 and 16. And that's coordinated by Sorbonne University. And the idea is to have reduced protocols, but still robust protocols that are adapted for sailors and that will generate genomics data as well as imaging data. Then we have activities with moored sediment traps, observatories, the blue stars on the map. So we will do genomics, plastics, and carbon analysis on selected sites and selected samples to cover time series of 10 to 20 years. And that's led by EMSO, NOC, AVI, and Plocan. And we will work with merchant ships. So you've heard all about the CPR, the Continuous Plankton Recorder. There's a huge amount of data in the North Atlantic from the CPR. It goes back 90 years. Now we will, in AtlantECO, initiate the first South Atlantic CPR survey, a line between Rio and Cape Town. We will sample it 4 times a year, starting in September next year. And on that CPR line, we'll do plankton and plastics and maybe genomics. And that's led by MBA and partners in South Africa and South America. And that's, yeah, a collaboration with merchant ships because the CPR is still behind merchant ships. And finally, we have technological innovations. The first one is an imaging sensor, the UVP, the Underwater Vision Profiler. It's developed at Sorbonne University in France. It identifies plankton and it estimates particulate flux at high resolution on the vertical. So in AtlantECO, we will test the deployment of the UVP6 on profiling floats. That's what you see on the image. And also on fixed observatories together with sediment traps and in aquaculture facilities in open water. And the second one is a genetic sampler. So the ROXY. So the genetic sampler collects samples and then they will be analyzed in the lab and used to develop lab-on-a-chip probes for the genomic sensor. So what we do in AtlantECO, we will test the deployment of the ROXY, of the sampler, on fixed observatories together with sediment traps, on the continuous plankton recorder or associated with it, and in aquaculture facilities as well. And that's a quick run through work package 4.
Okay, taking back the control, and I will pass on to Ferenc.
Okay, welcome to work package 5. Can I control? Let me see. It still doesn't work, but okay, it works. Let me go back. Okay, there's some delay. So, welcome to work package 5. We have 2 co-leaders of the work package. Ulani Makalanyane from the University of Pretoria, South Africa, and myself, I'm Ferenc Jordan from the Balaton Limnological Institute, Center for Ecological Research in Hungary. So I'm speaking from Hungary. Work package 5 is a work package which is of really synthetic and integrative nature. So it's like putting the little pieces of the puzzle together. And the pieces of the puzzle are the huge amount of information coming from the other work packages, the previous work packages. And here in work package 5, we try to put all these information together. We try to take a systems perspective, and the methodology is network modeling and network analysis. This is very important because, as what Daniela said, we are facing several challenges. We see many phenomena, we try to understand many problems, and these are not independent of each other. So all of them are the problems of the Atlantic Ocean, or the phenomena happening in the Atlantic Ocean. So these are linked together. And if we can have a chance to put together a network model, if we have a chance to think in terms of networks and systems at a large scale, we have a chance, or we start to have a chance, to study direct and indirect interactions, to potentially identify surprisingly strong or surprisingly important interactions between organisms and between processes previously potentially thought to be independent or not really linked to each other. So we are looking for hidden links and hidden indirect interactions between the components of the ecosystem. So a large amount of data should be put together in network models, and this kind of systems view can reveal all of these complex patterns. And actually, by systems analysis, by network analysis, we also have a chance to tame complexity, to handle complexity somehow. Otherwise, if you look at the network on the right, it's just not understandable at all. But if you perform network analysis, you can actually quantify the properties of these networks at the local level, the level of the nodes, where are they? At the global level, the level of the whole network, what to say about the topology, the architecture of the whole network, and also the mesoscale level, so what to say about the modules in the network, the little cliques, the little neighborhoods around organisms. So these kind of local, mesoscale, global approach to networks and these levels of network analysis can reveal a huge amount of information about interaction networks. Of course, not only in the ocean, in any ecosystem. But whatever kind of model we create and whatever kind of data we have and whatever kind of predictions we can make based on the modeling efforts, it has to be tested. Otherwise, it's only a lab or lab activity. So a very important challenge to solve is to try to experimentally test all of the predictions we take by network analysis. The more concrete goals, first of all, based on the local bits of observational information, sequence data, abundance data, presence-absence data, we need to identify novel algorithms for network construction, network inference. So the key of network analysis is to define and measure the links, the interactions, the effects between different agents of the same system. So to look for new algorithms is the first step. This needs computational ecology, this needs statistics, this needs machine learning, this needs mathematics. Basically, we are looking for statistical associations among little agents of information, and I would say between organisms or between sequences, OTUs or something like that. And if we can identify statistically significant positive and negative associations, then we can have the interaction network of positive and negative advantages, disadvantages, effects of different organisms to each other. If we use genomic data, if we use next generation sequencing data, then we can speak about OTUs. In other cases, we can speak about species or strains or maybe functional groups. It's also a matter of it's a matter of interest, and it's a crucial thing to decide what are actually the nodes in the network. And then we have to see what are the links in the network. So what kind of interactions we can speak about. If you have association, it's a kind of black box of different kinds of interactions behind. So the association can be there because of metabolic coupling. It can be there because of trophic interactions, predator-prey. It can be there because of competition, because of facilitation. So all kinds of the classical ecological interactions can be mixed up somehow. And then the final outcome of this is the positive or the negative association between organisms. If the network is ready, then we can use older and develop novel network indices, and we can use them to quantify and to characterize the network at several levels, as I said, global, local, mesoscale. And of course, we are interested in using indices that are relatively easy to use and they have high predictive power. So it's a very nice logical chain from the data to the final application in other work packages using indicators and predictors. So we have to use several network indices. We have to compare them. We have to compare the performance of these indices. And then we have to look for the best ones, where the best means that it's easy and easily applicable and predictive. Of course, again, the experimental model is very important here the experimental approach is very important for testing the models, for model testing, and we need to build up an experimental pipeline based also on onboard testing, onboard experiments, and also on aquaculture experiments. And in some sense, also network simulations, food web simulations are kind of testing the topological predictions of network analysis. On the right side, you see some visualization of this pipeline. So from the cruises, we can collect the data. Based on the data, we have network inference. We create the networks. Based on network analysis, we have predictions. For example, organism A has an influence of organism B, and then we can do these experimental testings for a couple or a few organisms. How do they behave together? So combined with nutrition experiments. For all of these activities, we need to have a diverse group of experts, and actually that is what we have. In Work Package 5, 16 partners are involved to some extent. Of course, the relative contribution is unequal. On the left, you see the 4 partners who are responsible for deliverables. So maybe in a sense, these are the key partners here. And actually the link of these, the list of these key partners shows very well the multidisciplinary nature of Work Package 5 and of the whole project. So first of all, CNRS has massive expertise in network inference, in the statistical methods, in the computational methods, in how to transform genomic data into ecological interaction data, and how to build the networks from this. So it's like computer science and genomics and the interface of the two, and statistical inference. The second is the Hungarian Academy of Sciences, has experience in network analysis, as I said, at several levels, using many tools for characterizing node level network properties and global network properties, so the mesoscale level. University of Pretoria has experience in experimental validation in an onboard experiments. That's a very essential part, otherwise modeling is just hanging in the air. And the Alfred Wegener Institute, for the fourth very important partner here, is doing research on indicators and to try to understand which predictors can be used later as indicators, how to apply all of the knowledge here, all of the predictions of modeling, how can it be applied for really solving the problems. And of course, all of the other partners will have important contributions, but based on the deliverables, these are the responsible partners. Activities, first of all, and I mean in terms of time, First of all, we need to work on the network reconstruction methods and the pipeline. We have to establish the methodology of how to infer interactions and how to infer networks, how to put together the networks. We want to work with cross-kingdom interactomes, and we have to create different kinds of networks different networks in space and different networks in time if it's possible. So we need to have a bestiary of networks in order to make comparative analysis, which is always very important in biology and ecology. Second, if these interactomes are created, and these will be like from the end of the first half year till maybe the end of the third year, the network analysis of these interactomes, and to try to identify the key positions in the networks, the key organisms, whether the network as a whole seems to be robust or vulnerable, what about the sensitivity of the network and on network positions as a function of their network properties. And then what is the most biological part is niche analysis to understand the redundancy, the functional overlap between organisms, whether or not they are replaceable. If one is going extinct, the other can take up the function. Who are the key players? Who are the most important organisms in the marine plankton system? And of course, we don't speak only about the planktonic system. If we have the data here, we try to extend and we try to extrapolate the results also in the direction of fisheries, as we have heard initially. And the experimental testing will dominate the middle and the second part of the whole project. Of course, we need something to test. So first of all, we build the networks, we do network analysis, and we are doing the experimental testing. That's it. Thank you very much for your interest.
Thank you. We'll now go on to work package 6 with Marcello.
Hello. Let's see whether it works on my side. Yeah, seems like, yeah, I got control. Thank you, Luis. Thank you, Daniele. Thanks, everyone. Good afternoon to all the participants and also to the Brazilian colleagues. That's finally afternoon for them as well. Thanks for being involved very early this morning. We'll continue with work package 6. That is also another modeling component of the AtlantECO project. It's about model predictability, and the use of the term predictability here is pretty important, because it's a fundamental requirement for any model that wish to become an instrument for ocean forecasting in this particular case, and also for predicting changes at the climate, at the global scale. If a model doesn't show predictability, that doesn't mean that it's useless. It's actually very useful from a theoretical viewpoint, but it can more difficultly be translated into immediate societal benefits. And I must say that we got an urgency here. So we need to assess consequences and take measures. Now, the particular challenges that this work package is facing are quite many, as you probably have seen. All work packages have been put together to face relevant challenges dealing with the Atlantic oceans. We do recognize one main challenge that is in measuring and describing in a mechanistic way the ocean microbiome. Particularly, we realize the chasm between the scales at which the microbial ocean functions and the system response that happens at larger scale. We got phenomena that are at much larger scale, like the planetary scale, dealing with the climate variability and the climate changes. So the figure you see here on your right comes from a very recent paper and a collection of articles that have been challenging and understanding more what are the issues underlying the difficulty of bridging together scales. So it shows the complexity of the natural microbiome and how it relates to the various ocean ecosystem components. Now, the microbiome lives in the turbulent ocean environment, and this is, as we know, and many physical oceanographers know, is nurtured by the larger scale general circulation features. And this creates regions of discontinuity and in some senses, some homogeneity, and all of them are connected by a multitude of scales. Now, human perturbations, they occur at multiple scales. but how they propagate physically and biologically through all these networks that we have seen or described, with in work package 5 is not really completely known. So we face a major challenge, particularly if we want to bring together the physics and the microbiology and the biology of the functioning of the ocean. So we started working on this topic about 70 years ago, and we are yet to find an equivalent of a thermodynamics theory that would allow us to describe the ocean microbiome with a few state variables. We did try. We've been trying to put together the most relevant variables and indicators that we thought that were good enough to represent the system. But I must say that our predictability is still lagging behind. Nowadays, we got growing computing resources, we got an advancement in observational techniques, and we got some new knowledge. So it's very much time to find that those advancements find a way into predictive models. So to do this, we put together a team with a series of goals. Now, on the right, you see the very starting point of this work package, and the availability of high resolution and quite realistic simulations of motion in the ocean. Now, our main interest is to quantify and reduce as much as possible the uncertainties in the physical connectivity between the different biogeographic regions and in what is going to happen in those regions. To do that, we need mechanistic descriptions of the functioning of the microbiome. And this can be done by improving our existing biogeochemical models, taking advantage of this growing body of information from augmented observations, organic matter fluxes, imaging, and all the omics that have been introduced earlier. Having done that, we need to demonstrate predictability somehow. We need to check whether these revised formulations are actually better in demonstrating and in resolving the processes that we are slowly elucidating in terms of some major ecosystem services like the carbon export, or to see whether there are going to be changes and shifts in community responses and how multiple stressors would be working together in favor or against. Unfortunately, in most of the cases, what we've seen are negative impacts of all these combined stressors. As the team we needed to put together, sorry, I was supposed to be the previous one. The team that we put together is composed of 13 partners. One of them is an external partner. They are experts in physical and biochemical modeling, and we have been teaming together with the other work packages that they are very much represented in this work package in terms of ocean observation. So the kind of instruments which are knowledge instruments and real instruments that are combined together in this work package are the high resolution ocean modeling and transport, the development of new functional and mathematical parameterization of biochemical processes, and the use and the translation of information from multiscale and multidisciplinary observations of the ocean in terms of images and genomics in such a way that they can be incorporated in models. Now, this means that we just don't need the data. We need the data originator who knows what kind of data are being created and how they can be used for improving the mechanistic functioning of these models. We will also take advantage of what's going on in the US, and particularly in the University of Maryland group that's going to team with us, with all the partners in the project. Now, physical connectivity is one major activity. You have seen already a simulation. Sorry, it's a bit slow in reacting, so I'm quicker than that. You have already seen a simulation earlier about the global ocean. We will be using data from 116 degrees resolution, which are approximately 5 to 7 kilometers in the global ocean, and you can understand how much computer power is needed to generate these data in 3 dimensions, so not just at the surface of the ocean, but the interior. And what we are usually showing is just the surface of the ocean, but the microbiome is everywhere, also in areas that are not illuminated, where the remineralization processes are occurring. So we want to resolve all the physical drivers at multiple scale. We want to start from the global ocean, the Atlantic Ocean, and goes to the scales where human perturbations are more likely to occur and to be visible as we get closer to the coastal ocean. So knowing the scales of the seascape is essential. And we're going to do it through a combination of Eulerian and Lagrangian formulation. So one describes the oceanic fields, the Eulerian formulation, and the other one, the trajectories of elements, parcels, and their content in the ocean. Lagrangian oceanography is very much a keyword in this work package. It's going to tell us what time scales and how, through which pathways, water would flow from different locations and biogeographic regions in the Atlantic, and with that, how water pollutants, plastic, plankton, and other organisms are transported. But how do they interact? Well, in that case, we have to move to a microbiome predictability, which means we have what you see here is a rather complicated representation for actually a complex problem. And it's not at all a linear problem. I mean, the presence of an arrow here doesn't mean that we know exactly where we're going. That's why there are wiggles on these arrows. And it goes from augmented observations, so the availability of data we got at the moment, to the global scale and the problems that we would like to resolve and face. Now, how we get there is through a combination of existing knowledge and new theory that we are working on. Particularly our interest is the parameterization of how the microbiome operates at the cellular scale. For instance, how co-limitation and interaction between nutrient availability, micronutrient availability, light availability, and all the other drivers of the function of the microbiome, they interact together to provide the primary production and the major functional features in the microbiome. And through that and the interaction with the physics, which can be done in different ways. So up to date, the Eulerian approach is the one we've been mostly doing. Now we're moving towards Lagrangian approaches, Lagrangian examples, and new ideas are coming out, which will be further developed into this work package. So our hope is to finally get to an improved understanding and a more realistic presentation of the microbiome functions and how it would respond to the future environmental, well, the ongoing and the future environmental fluctuation. And this will be done in collaboration with the other work packages that will be coming after, presented right after me. So I thank you very much for your attention.
Thank you, Marcello. Now we'll go to work package 7 and handing over the control to Bruno.
Yes. Can you hear me? Okay, I think I got control now, maybe. Let me check. Okay, then my name is Bruno Bongiorno-Ardelli. I'm working at the Italian National Research Council, and I will be co-leading WP7 together with Willie Wilson by the Marine Biological Association in Plymouth, United Kingdom. And well, the WP7 is actually introducing a different approach to several of the questions that have already been put on the table. Actually, we have seen that AtlantECO will be providing some really unprecedented level of details about the marine ecosystem and organisms in the Atlantic through very innovative observations and incredible collection of new data. And we've seen also that there will be considerable amount of effort dedicated to developing new models, and I would say the network models, they are meant in a very different way with respect to what a classical modeling is thinking about usually. And Marcello just showed how many question marks are still present in the modeling of the biogeochemical oceans, and how they plan to make a step forward in improving this modeling. WP7 will be looking at things in a slightly different way. From some point of view, it might look like a more classical approach, but putting the, let's say, time evolution as the key aspect of the work package. So the idea is to try to observe from what we have, what is the true drivers of the ecosystem changes that we can observe in our observations and reanalysis from models. So basically, the challenge will be to identify the dominant patterns of variability and try to associate them with processes that drive the marine ecosystem changes, and and also to detect extreme events and eventual regime shifts that we can find in these records. Why is it a challenge? The reason is very clear to, well, even clearer after having seen some of the previous presentations, because the system is extremely complex and there's very different kind of dynamics that characterize each of the sectors involved. I mean, biological life maybe on Earth lives in something that is evolving more slowly, a landscape that is more or less fixed. But when we think about the oceans, we have currents, and it is actually truly a seascape that we have to think about. And observational data, however, they cannot cover all the variables that would be needed to get a complete description of the state with the right level of space and time coverage. And on the other hand, models, they also have problems because they need some data to start from. So there's problems with the initial conditions, and some of the processes that the models try to describe, they're not really well known, or they need to be parameterized when going, especially when going at the unresolved scales. Then there's a problem by itself. It is a challenge to face this kind of questions, because the system is by definition nonlinear, which means that we usually think or try to think about processes and try to analyze processes in a separate way, but these processes may interact, they do interact, and they interact in non-trivial ways. And one of the factors that is impacting at it, and which is not really well known or easy to describe quantitatively, is the stressors related to socio-economic activities. So it is a challenging question we are facing, but of course we have some very practical goals we will be trying to achieve. So the first one will be very easily to assemble state-of-the-art time series datasets. So we are focusing on the evolution over time of what has been observed in the past of ecosystem drivers and stressors. And then we will try to identify the processes, both environmental and ecosystem processes, and their patterns of variability, by combining some kind of more or less classical statistical analysis and innovative tools like machine learning, or more classical Eulerian-like analysis of what we can observe in the data. And then there will be, of course, a goal to identify which are the most important stressors and what is leading to extreme events and eventual ecosystem regime shifts. And considering not only the environmental drivers and stressors, but also the socioeconomic ones. Then in practice, what we will be doing is exploiting what has already been produced as a result of big efforts by mostly by the European Commission in the last few years. So we will be on one part of the work will be focusing on 2D and 3D reconstruction based on observations of the essential ocean variables and of biological proxies that are provided by the Copernicus services, specifically the Copernicus Marine Environment Monitoring Service and the Copernicus Climate Change Service. And one important point is that the data we will be looking at, many of them have been produced in the last years by the people involved in the work package. So here it is where people who have devoted a lot of effort improving 3D and 2D reconstructions of the oceans, it is where we all aim to analyze this data and give a better description of the variability and processes affecting the Atlantic. But we will let's say, complement this information with more detailed information we can get from time series of in situ data that again have been produced, collected, and even already organized and analyzed in the past by partners of the team. So we will get into more details because, of course, 2D and 3D reconstruction of global ocean and of the Atlantic Ocean, the ones that we will be looking at, I was introducing before, they cannot provide much details on the biological and biochemical variables of interest. So here we will get more details, but it will be less, a smaller coverage, spatial coverage. And then, of course, we will also have the opportunity to analyze the new models that have been introduced by Marcello a few minutes ago. Sorry, again, oh, it's very slow. So, for example, there will be analysis dedicated to the physical component. So we will exploit the 1/16 degree CMCC analysis, but also dedicated efforts to analyze the interactions that can be described by fishes, which has been developed by one of our partners. And of course, we will exploit all these data, sorry for the delay, by applying different kind of analysis. As I said, we aim to identify the dominant patterns of variability at first, looking at regional modes, global modes, teleconnections, and this will be done also exploring new tools like nonlinear principal component analysis or similar machine learning approaches. Of course, we will be looking at trends and extreme events and trying to identify eventual regime shifts. And we will also be focusing on key controls of ecosystem services. And for example, there will be activities specifically dedicated to look at the subsurface fluxes of particulate organic carbon, also discriminating between sinking and subduction effects, and on biodiversity, where there will be a very strong link to the species distribution models that will be developed within our package 2. To carry out all these activities, of course, we will be working in collaboration among, oh, sorry, this is so slow, among partners that have very, very complementary experiences. The team is very qualified in several aspects, when going from the collection and processing of biogeochemical observations to the design and development of advanced models and observation-based products, but also on the combination and analysis of complex data sets. So it will be fun, it will be difficult, but we look forward to start working on the data within AtlantECO. Thank you.
Thank you very much, Bruno. And now we'll be moving on to work package 8 and handing over the control to Marion.
Hello, everybody. I hope you can hear me. I'm just testing. Eloise, I don't know how to move the slides. Eloise, okay.
I can do it for you if you want. If you just tell me next slide, then I will.
Yeah, because okay, yeah, it doesn't go from my computer. So the title slide. So no, the first slide. Sorry. So hello everybody. So my name is Marion Gehlen. I'm from the French Commission for Atomic and Alternative Energies, and I'm leading WP8 predictions of and for ecosystem services together with Carlos Lentini from the Federal University of Bahia. I don't know whether you see me, so it's a stupid question because I don't see myself actually. So now coming back to WP8, many things, challenges and aspects, thank you, have already been addressed by the other speakers, particularly by Marcello and Bruno, because as the preceding two work packages, WP8 is also the work package that is structured around modeling. Next slide, please. So very much of all of the understanding of the dynamics of ecosystems, the drivers behind the ecosystem dynamics, as well as the controls of the structuring of the ecosystems, and the way these aspects can best be implemented into models. All these efforts gathered throughout the project will ultimately feed into WP8 to achieve improved predictions of and for ecosystem services. So the challenge of WP8, to put in a single sentence, to my understanding, will be to progress from physical biogeochemical projections as those that are done routinely, for example, for the IPCC exercises, towards projections of the dynamical Atlantic seascape. This implies to integrate across physics, chemistry, and biology in a much tighter way than what is done as for today. The figures that you see on the slide here show the potential of such an integrated approach when it is extended to a novel data set. In this case, I'm talking about the genomic provinces that is the novel data set that has been integrated to information on the physical chemical environment to produce the first ever biogeographies of the marine microbiome at the scale of the whole ocean. So this is just an example of what can be done. And it shows it's a first step towards improving our capacity to project the future of the Atlantic seascapes. So WP8 has a scenario-based approach, and it will provide input to ecosystem-based management strategies. And also, it will also project indices of ecosystem status, ecosystem health, for instance, and also ecosystem services under different scenarios of climate change and also socioeconomic pathways. Next slide, please. So the goals of WP8 are, at one hand, to predict the future of basin-scale Atlantic ecosystem processes and to evaluate the likelihood for ecosystem tipping points and to regime shifts in response to stressors when following different alternative scenarios. We also aim at predicting and quantifying changes in ecosystem services, and the services that have been identified are carbon sequestration, nutrient recycling, primary production, and this in response to high and low emission climate change scenarios. Next slide, please. So the team behind WP8 is, as for all the work packages that we have been listened to, seen this afternoon, we also have a very interdisciplinary team. So it's a unique combination of expertise in WP8. It relies on three expertise pillars, so to speak. It connects the physical, biogeochemical dimension, where we have experts in climate change projections, in impacts on ocean physics and on marine biogeochemistry, impacts of climate change on ocean physics and marine biogeochemistry with partner CEA, so my own home institution, Cape Town, Naples, and Brazil, the University of Bahia. The next dimension is the biological ecological dimension, where we have experts in studying of the impacts of climate change effects on communities, ecosystems, as well as key processes. ETH Zurich, CA, the Napoli Station, and University of Liverpool. And finally, we also integrate with the economical dimension. And that's a really challenge too, to translate the impacts that we have quantified in the physical, biogeochemical, and ecological dimension, to translate this into indicators of ocean health and ecosystem services, and finally to feed this into macroeconomical assessments. There we have CMCC, OGS, and USC. So all these three dimensions will feed together to provide improved assessment of the future status and the dynamics of the Atlantic socio-ecological system. Next slide, please. So how will we now reach these objectives? So we have two approaches, or actually it's one approach, but we are using two types of model systems. So the first one is the projections, so the projections of the dynamic Atlantic seascapes. Here we will combine the output of Earth system models, which provide the evolution of physical biogeochemical stressors in response to different shared socio-economic pathways, the SSPs that have been developed for the ongoing IPCC assessment report. So the stressors will be taken out from this Earth system model projections, and they will force two types of models. The first one will be developed under WP2, as exemplified here on this upper panel. We have a species distribution model, which in this case relies on the collection of several hundred and the analysis of several hundred of plankton species and present day and also in the future, under future conditions. So this approach will allow to evaluate impacts of climate change on biodiversity, just as an example. Another type of models that we will be using as the more classical mechanistical model, and these models are actually a part of the Earth system models that are shown, for which we see the results and projections here. One such model is the PISCUS model that is indeed developed at IPSL, so again at my home institution. But within AtlantECO, this model will be augmented at the University of Liverpool by extending the scheme of nutrient limitation to include several other micro elements or micronutrients. So this will then allow also improved prediction on the future evolution of, for instance, net primer production with this model. So all taking this together, the objectives are, or what we will achieve, is we will have an improved representation of the variability of ecosystem drivers and the detection of regime shifts under future climate change, and improved prediction of the Atlantic ecosystem change, as well as improved prediction of ecosystem services. Again, the three key services that I mentioned before: carbon sequestration, nutrient recycling, and primer production. Next slide, please. The other strain of activity is, from my perspective, from my own scientific background, even more challenging, because we will move towards socio-ecological projections. For this approach, we will again combine the evolution of physical biogeochemical stressors, so derived from Earth system model simulations presented before, but this will also be combined with the evolution of socio-economic stressors that will have been identified in WP3. The challenge here is to translate impacts of climate change on ecosystems into change in selected ocean health indicators and ecosystem services, to combine this with the specification of kind of scenario drivers, but now we have augmented scenarios. For example, for fisheries, this would be the price of fuels for fleets, the population, the human population, the GDP growth, and the GDP and the growth for demand. So these two information streams will be the input to economic models, which then will output the evolution of GDP production and prices in response to these scenarios. This allows to explore impacts of different climate and management scenarios on ocean health, as well as to explore impacts of different climate and management scenarios on ecosystem services. And we think that this is a very important contribution or information to be handed over also to decision makers when it comes to shaping new policies and environmental policies with the aim of keeping a sustainable Atlantic Ocean also for future generations. Thank you.
Thank you very much, Marion. And now we will move on to work package 9 with André. André, do you want the, sorry, I have a cat. I don't own a cat, but there's a cat in my house.
Yes, hello all.
Do you want the
yes,
control?
I can take the control.
Change the slides.
How do I pass the slide? Just to click? Yeah, if you can do it, it will be simple. So hi all, Eloise, please, I will indicate you to the next slide. Thank you all for this, this scientific great overview. I'll be more simple on our communication and outreach issues. I'll try to be quick. So first, the challenges we have, of course, building the All Atlantic community based on the Belém Declaration. So we were there in Portugal for this inspiring event on the launching of the Belém Declaration. The idea of the Commission, Brazil and South Africa governments is really to promote this All Atlantic spirit with an All Atlantic community aware to the challenges for the Ocean Atlantic. So first challenge for us all is to translate the scientific issues for a broader audience. Microbiome, seascape, microplastics are not so easy to communicate to people beyond scientific audience. So for us, it's a real challenge to find the entry doors and the appropriate language to communicate on microbiome, on seascape and others. And then create a network of stakeholders around the Ocean Literacy Platform. We are lucky we have with us in the consortium the UNESCO IOC. Francesca Santoro is with us for co-leading the work package, and Francesca is also the coordinator of the Ocean Literacy Platform with UNESCO IOC. So it will be an important challenge and important activities of the project to make it real, to make it dynamic and connect with the regional hubs in South Africa and Brazil. And of course, promote capacity building with concrete actions, transfer of technology, protocols, and etc. We'll be working with the EMBRC, with UFSCar and others, and particularly with synergies with WP2 and WP4 for this capacity building. Please, next slide, Louise. The goals, more specifically, some words about the tools we are building for the site. So the website is online now. Thank you, Eloise. Thank you, Daniele, for helping, for co-building this with us. We are now preparing the branding on social media, preparing a launching movie for the whole projects. We are also of course, preparing a poster in a folder to present the project. The second main goal is to structure and share knowledge. As I said, it will be in synergies with WP2 and 4 mainly, but also with WP5 and 6, and more broadly with the whole consortium. It's an important step for the project, the whole capacity building stuff around, and then promote ocean literacy. And I'm happy we had a meeting with Francesca and with colleagues, and the idea is to structure an ocean literacy strategy for the Atlantic. So the ocean literacy platform had an experience in the Mediterranean around building a specific ocean literacy strategy for the Mediterranean Sea. And the idea is to take advantage of the project and to build a strategy specific for the Atlantic, of course, connected to the Brazilian government, to the South African government, to the local committees already established. There in Brazil is quite active, the ocean literacy stakeholders that are already working around this concept. But again, it's a challenge to make it for all. Ocean literacy it's difficult to translate in other languages, in Portuguese, in French, and so it's also a big challenge, but an important goal for the project. After next slide, please. The team. So the Tara Ocean Foundation, we have 15 years of outreach with the Tara Schooner. Tara Ocean Expedition was already a big challenge to promote plankton ecosystem to a large public, and we did it, I think, brilliantly with the scientists working together with media people, communicators, artists, and the idea is really to communicate beyond the scientific audience, and the fact that we have 3 sailing boats in the project, and especially with the Tara and Eco sailing together, the port calls in rivers, so lots of opportunities to communicate and to reach, to have a broader impact with the port calls and these boats, which carries the story of the project. As I said, the UNESCO IOC is with us. The Ocean Literacy Platform will be a tool for all. Also, the Ocean Literacy Platform is linked to the Decade of Ocean Science and for Sustainable Development that will be launched next year. So it's quite timely for us this decade. Also, it's kind of challenge for scientists. And the 7th pillar of the decade is actually the ocean literacy actions. So really connected with the decade and the ocean literacy, thanks to UNESCO, IOC, and the team. And we have the regional hubs. So for Brazil, it will be the University of Santa Catarina, as I said, with the Eco boat. They had already developed very nice outreach on plastic pollution for the launching of the boat. So there is interaction with sailors, with surfers. Santa Catarina and Florianopolis, very nice place to promote these interactions. And the University of Cape Town with Marcello will be our regional hub in South Africa. And of course, I don't need to tell about the large experience of outreach for ocean sciences of the Cape Town, the link with 2 Ocean Aquarium, blah, blah, blah. So next slide, please. Just some words on the activities, the main ones, because there's a lot to be built together. First, to manage the website and social media with strategies for different targets. I mean, there's not only have a Facebook account and have an Instagram account, you have to strategize all this communication to think about what kind of public need, what kind of messages. So it's not only a website and social media, but there's a strategy behind it. It's very important. As I said, one of the activities will be concentrated, will be joint on this Atlantic Ocean Literacy Strategy that we will build. It's a very nice proposal from UNESCO IOC to build this platform within this project, and of course with some synergies with Anchor and the sister projects for sure. And then produce outreach materials on microbiome and microplastics for youth and scholars. Young audience and youth are a target for the project, of course, and a way to build this community for tomorrow. And last slide on the activities, please. Please, the next slide, Eloise. So, as I said, the port calls of flagship boats, AtlantECO, will be an opportunity, and we are coordinating with ECO since now for a more impact, a stronger impact we can in this port calls. Of course, the other flagship boats, Eugen Siebold, Alpha Crucis, and RS Agulhas, will also be using the same tools and the same branding, of course, but we are specifically focused on AtlantECO with the rivers for a broader impact, for extended impact. We also intend to develop partnerships with media groups, mainstream media corporations, not only to stay with blogs and nice specific websites, but trying to go beyond the scientific communication, trying to reach an audience. We have quite a lot of experience with Tara to making movies and working with artists, and I think there's a great opportunity in this project to link all this incredible science that will be produced with some media group that will be interested in help us to tell this story. So one of the activities will be to produce a web series telling the story of the All Atlantic program and telling a story with a red line to explain to different targets the ambitions of the program, but also linking with the territories, priority zones, arriving in territories and trying to link with local issues, linking with local communities, fishermen, aquaculture, etc. We also planned an activity on an exhibit on plankton microbiome. This will start with more simple version, but the idea is to work with all work packages and the whole consortium to pass from a simple exhibit to a more ambitious one to be proposed to museums, to aquariums, and to a more broader audience. And I'll stay here. There's a lot to do to communicate together. Thanks.
Thank you, Andre. Now moving to our last work package, which is work package 10 with Pedro.
Yes. Hi, hello everyone, and welcome to work package 10 for data management, dissemination, and exploitation. Louise, I don't know if I can change my slides or...
Do you want me to do it, or do you want to have control?
If I can have it, yeah. So, thank you. As you can see, this work package is managed by SPI, myself, Pedro Campillo, and also Sintef with Alexander. I'm not sure if the slides are changing. No, yes. As a challenge, we have the first challenge to ensure that all the data that is generated by AtlantECO is findable, accessible, interoperable, and reusable, so it complies with the FAIR principles. Also, that is available in open access, but also in balance with industrial exploitation. Another challenge is to ensure the maximum uptake of the results, and also to engage in exchanges with the stakeholders to maximize the impact of the project. As goals, we have these three main goals. The first one is to develop a data management strategy. Because in this project we will have a lot of different data in enormous quantities, so we have to have specific guidelines to know how to store and how to disseminate all this data. The second goal is to have a dissemination and exploitation plan to make sure that all this information that the project is going to generate is going to reach the relevant stakeholders. And also to develop policy briefs that include, of course, recommendations to tackle all the challenges and complexities of the marine. The team. As I've said before, this work package is led by SPI, which is an international consultancy company from Portugal, and SINTEF, which is a research organization from Norway. We have here the splits among partners. First, for data management, we have the European Molecular Biology Laboratory, EMBL, that is going to be is going to have a very crucial role here in this data management section. Also, Seascape from Belgium, UNESCO, and Sorbonne University. It's important to refer that these four partners, beside the SPI, they work very closely with several data resources. So they are going to be crucial in this data management process. Also, the partner EMBL will be very important because it's going to be the facilitator between AtlantECO and all of these data access resources that these partners are going to provide. So these are partners with a strong background on marine science, and that they develop high quality data services. Regarding the case studies, the partners involved are going to be SINTEF, the University of Bahia in Brazil, University of Cape Town, and also University of Santiago de Compostela in Spain. As the main activities of this work package, we have these four pillars. So the data management and the dissemination and exploration of results, the contribution to the marine strategy and policy through the policy briefs, and also finally, but not least, the case studies and the roadmap. So regarding the data management, as you can see, we have a very different kind of data that's going to be produced and organized in this project. So environmental, genetic, imaging, socioeconomic, and also maps. And since the AtlantECO is a data-driven project that generates a lot of data, we need to have here a process to see how to integrate all this massive amount of data and how to store it and disseminate it also. So we need to define here protocols and rules to know how to deal with all of this data. The partners of the project are going to provide this kind of services within AtlantECO, so the data creation, annotation tool, and data portals. As you can see on the right, we have the four partners that are going to be mainly involved in this data management, and also the quality data services that they provide, such as data archives and dissemination portals. So in this work package 10, we need to have an ambitious data management plan to deal with all of these data and the preparation of data and metadata, and how we can archive it and put it in the dissemination portals. It's also important to refer that all the project outputs are going to be stored at the European Digital Repository Zenodo. So, as I said before, we are going to have a key stakeholders database that's already identified as main contributors, the policymakers and industry, but also, of course, another kind of stakeholders, such as representatives of academia, international organizations, and the civil society. Regarding the dissemination and exploitation plan, this one is going to be built in order to see how we can disseminate the achievements of the project to a large scale of stakeholders and also targeted audiences. Also this plan is going to identify the key outcomes of the projects, and also of course opportunities identified also for future research and development, and of course commercial exploitation. Within this work package, so we are going to also produce some policy briefs with the policy, legal, and institutional measures, including, of course, recommendation in all of these areas, and in very different levels, so national, European, and international levels. Also, we'll have some case studies, 5 case studies, as Daniele presented in the initial presentation that he made. And finally, we will have a roadmap to determine how the future activities will exploit the project results. And regarding work package, and it's all. Thank you.
Thank you very much for this overview, very exhaustive overview of the different work packages. As you have seen, the project is very rich and complex. Now we have a little bit of time for a few questions.
So the question was about the blue cloud. The question was, will we use, in AtlantECO, will we use the blue cloud? So the blue cloud is part of the EOSC, the European Open Science Cloud. And the first element of answer, partners from AtlantECO, so EBI, Sorbonne, CNRS, and CEA are leading one of the demonstrators in the blue cloud, the plankton genomics demonstrator. So we are extremely linked to the blue cloud in AtlantECO. And the answer to the question is that the blue cloud probably won't be mature enough in the first 2 years of AtlantECO so that we can use it. And AtlantECO is assembling lots of data that are probably not available yet through the blue cloud. So we won't be using it initially. The idea, of course, is to make all the data that we collect in AtlantECO, make it available through the blue cloud eventually. And the modeling, so for example, niche modeling that we develop in work package 2, would be deployed in the blue cloud. So we will use the experience that we have at the moment with the demonstrator, learn how easy it is to deploy a code, and that should be easier for us to do it.
Hi, this is Nicola. Thank you, Stefan. I think you hear me, right?
Yes, I can.
Wonderful. Yeah, thank you very much. No, what I wanted to say is that Blue Cloud is not so much a portal where data products would be provided. It's more about providing the tools to federate very different databases or data infrastructure and provide the cloud computing and services, analytic services to be able to exploit all these different data sets. So it would be, I think, important to ensure that, because I understand you're doing pretty much the same here. It's aggregate very different data sets. So it would be good to ensure that if you're not using similar tools that are being developed under Blue Cloud, that they are compatible with the framework that they're developing, that they can complement, or probably you can also reuse some of the things that they are just developing now. So I think it would be interesting to have very early contact with them and see how to best maximize the cooperation between the two projects.
Yes, and as I said, it's the same partners, the same people.
Yeah, and that's very good.
Yes.
That will definitely facilitate.
Yes, so the plankton genomics demonstrator will put in some niche modeling code pipelines in the Blue Cloud. But what is developed in AtlantECO, for example, will include connectivity, which is not included in what the demo is building at the moment. So we will build on the experience of the demonstrator, but what we will deploy is more advanced than what will come out of the Blue Cloud demonstrators.
Yeah, that's perfect. So I just wanted to make sure that the underlying technology to aggregate the things is easier, because this is where we really want to go with regard to data, is to be able to get data from very different sources quite easily, as far as easily is possible, of course. But I think there's a very interesting cooperation here.
Yes. And as you saw in Work Package 10, so data products will be deployed, well, archived and made available in EmmonNet, Eurobis, and Obis, which feed into the Blue Cloud. So yeah, we're in a good alignment.
When we had the meeting with Anchor, we talked about the idea of having an Atlantic JS portal. So that would be also a source where the data product could be provided through.
Unmuted. We have a question on how Work Package 2 and Work Package 10 are linked with the data sets. Maybe Stefan?
Maybe I should. How they are linked with a key partner. So we have Seascape Belgium, who they are hosting the Secretariat of EmmonNet, and they are linked to many European projects, and they develop what, well, sort of a portal based on GeoNode. So that's our link. So Work Package 2. So Seascape Belgium is part of Work Package 10 and Work Package 2. So they will make sure that all the products developed in Work Package 2 are already formatted in a way that they can be imported rapidly into the GeoNode. So yeah, and then the GeoNode is our basis to disseminate that into EmmonNet, into the European Atlas of the Seas, for example. So that's Seascape Belgium is the key partner to link Work Package 2 and Work Package 10.
Okay, we have a next question for Work Package 9. Are there any plans to engage the AORA Galway Ocean Literacy Working Group? This might link to the US and Canadian colleagues.
I can take this one. Yes, sure. Firstly, through UNCO, which is kind of the next generation for AORA. But also we've been participating on the our working group on microbiome with Stefan, Daniele, Pierluigi, and others. There are already some links on the microbiome working group, but yes, we will try to engage with this ocean literacy working group with our, but maybe it will be more through the ANCO joint actions and synergies.
So there was another question on the chat on the time of the data. How backwards should we go? Maybe I can repeat my answer. So the genomic data is really recent, so that's not difficult to set a limit. But the microscopy, there are data, as Mikey told me on the chat, she went through the books in Russian to extract data from the 20s. We're not going to do that, but we're going to gather the data that is available. We're not setting a limit of time if it's old data, but especially we'll put more emphasis in the last 20 years, probably.
I think a focus period, as far as I also understand from our discussion with Stefan, is the last 10 to 20 years. If someone has relevant data, we'll make sure we try to include it.
Well, not exactly a question, just a clarification that comes or follows discussions between Nicolas and Stefan, and also what Andrea Abreu just said on the ocean literacy. So regarding the Blue Cloud and AtlantECO, AtlantECO has already sent several very useful ideas for the anchor joint action on data. And because of that, there are now ideas to link with AtlantECO and the ModNet. So the synergies are already come out from all these projects, which is very good. And regarding the ocean literacy, our joint action related with the ocean literacy aims to build an all Atlantic blue school. So it will not really discuss ocean literacy in general, but the aim is to implement a tool of ocean literacy, aiming especially to the young people and their teachers. But you will hear more about it in the next presentation after the break, I believe. But this was just to clarify. Thank you.